Hello, everyone.
Hello, ma'am.
Welcome to to session one, the ethnobotany session, where we have a number of speakers with us. Today, we are going to start with Mr. Idris, and I will hand over to him with his talk on the phytochemical profile of extracts of Bidens pilosa and its correlation with cell proliferation, antioxidant, and larvicidal activity. Okay. Edward, if you could. Okay, thank you. If you're ready to
Thank,
go,
all right.
Oladeo, you can start.
Okay, thank you for having me. Just a moment, I want to share my slide. Oh, not yet. Just a moment, please. Okay, can you see my slide?
Yes.
Oh, we are fine now. All right, my name is Olade Ahmed Idris, and I am a postdoctoral fellow at Northwest University. In my team, we're actually working on biopesticides and the efficacy, as well as the impact on the environment. So, but I'll be presenting a part of this research, which is titled Phytochemical Profile of Water and Ethanol Extract of Bidin pilosa and its correlation with antioxidant activity, cell cytotoxicity, and larvicidal activity. So I'll be taking you through these follow outlines as we go by. I think it's necessary for me to start with the background of medicinal plant and their general uses. So medicinal plant has long history of being used in the ancient Egypt, China, India, Sri Lanka, and some part of Europe, and it has been used for different ailments. And medicinal plant parts such as the fruit, the leaf, and the root has been used so far and reported so far, and it has success in this regard. So however, medicinal plants usually have some few properties which enable them to be used for the treatment of diseases. For instance, it has the antimicrobial properties. These antimicrobial properties enable these plants to be used in the treatment of fungi, bacteria, even viral infections. Some of them has these antioxidant properties, so it enables them to be used in the treatment of wound, in detoxification, and as well as inflammation diseases. Medicinal plants are not only they are also used in the treatment of diseases in livestock as well, which otherwise called the ethno-veterinary medicine. So it is in most cases used in the treatment of ecto and endoparasites such as tick and parasitic worm. Some medicinal plants are also used in weedicide. So because they have this allopathic effect, which allow them to be used in this regard. However, our study based on Bidin pilosa, which is also a medicinal plant. It has most of the characteristics that I mentioned above. So Bidin pilosa is also called blackjack in the general name. It is an annual weed that is widely distributed in the tropical and subtropical area around the world. It can grow up to the height of 30 to 100 centimeters tall. On my left-hand side, you can see the picture of Bidin pilosa, and you can see the area part, the seed and the flower. Some of us might be used or familiar with this plant because of people who love to hike in the bushy area. Their clothes or shoelace might be stuck with the seed of Bidin pilosa as a result of the trichome that it possesses. Now let's look at the general uses of Bidin pilosa. It is around the world. It is being used as food, traditional medicine, biopesticide, and herbicide at the same time. And let's look at the medicinal use. It is being used to treat hyperglycemia, malaria infection in Southern Africa, tumor, microbial infections, and rheumatoid arthritis, inflammatory diseases. It is also used, it has been reported to be used in the treatment of contaminated soil with heavy metal, and it has also been reported to be used as a plant biopesticide. Now, looking at the general uses of Pilosa, you can see from one side that it is being used as food and medicine at the same time. At the other side, it's being used as biopesticide and herbicide. So it's like consuming what you use in killing pesticide and herb at the same time. So this brought about a question in our study, and it brought about the question: how safe is Pilosa? Apologies, it's not coming up. Are you there? Oh. Sorry for this. I'm not sure what is happening with my slide.
Okay, just click on it.
Okay, allow me to restart the program maybe.
Yeah, let's stop your share and let's restart it.
Oh, sorry about that, please.
Sorry?
Now I'm trying to, pull up the PowerPoint once again.
No problem.
Okay. Okay, can you see my slide now? Okay, sorry about that. Apologies.
No problem. So you now,
okay, thank you.
You're now on the introduction.
Yes. All right. So the use of Pilosa actually brought about 3 questions that lead into this study. So, and the questions are: what are the health risks of consuming Pilosa? Then the other question is there any correlation between the phytochemical of Pilosa and the biological activities? So we decided to answer this question following these materials and method. Firstly, we collected Pilosa from Lipidilele local municipality in Limpopo. So the plant was authenticated, and some of the samples was actually deposited in the herbarium, in the graphene herbarium at Noordwes-universiteit. So we now decided to, extract the active component of the plant using 2 solvents, which is the distilled water and ethanol extract, which is a common solvent being used among the local people. So the first thing we did is that we pulverized the plant after we macerated the plant, and the filtrate of the macerate was now kept in the refrigerator at minus 86 degrees for 24 hours. Then after this filtrate was then lyophilized in a fish dryer for about 5 days. So the samples, the extra that we got there was now kept in the refrigerator. The first analysis we did was the antioxidant content and antioxidant activity. So the antioxidant content and activity was now done using the spectrophotometric method. So we started with the total polyphenolic content and total flavonoid. The total polyphenolic content was checked using the standard toluene cicature, while the total flavonoid was done using the standard catechin. So we now extrapolated the result of what we got from this. Likewise, we decided to test the scavenging ability of our samples using the ABTS. Likewise, we also tested the reducing power ability of the sample as well. So we move to the next assay, which is the larvicidal activity. So the larvicidal activities was used to test the potency of the plant against the parasite, which is drug-sensitive parasite, the Plasmodium pisciparum. So the Plasmodium pisciparum was maintained in 5% red blood cell in RPMI medium, and also maintain a gaseous environment, which is 5% CO2, 5% oxygen, and 90% nitrogen. So we then carry out the tests in this regard in a 2-point concentration, 10 microgram per mil and 20 microgram per mil. So we move to the next assay, which we decided to test the toxicity of the extract, both of the extract, using this excelligence system. So the excelligence system allows us to test how potent or how toxic the plant extract is within 24 hours, unlike the regular MTT process that checked after 24 hours. So we're able to draw a pattern in this regard as well, following the system that was able to, draw a pattern from the response of the extract with the electroplate microplate. So after we decided to quantify and profile the phytochemical as well using the HPLC DAD. So after the HPLC DAD, we did the LCMS using Quitof. So in this SCMS, the SCMS analysis, we try to quantify what we have by using about 15 standards which were mixed together, and the standard were then put into various concentrations where we're able to draw a linear calibration curve that allows us to quantify the samples. Then the raw data was now analyzed using MZMI. Please allow me to pass this slide because of time. So I'll be passing this slide because I think I've explained this using the schematic diagram above. Likewise, this one. Then I'll simply move to the statistical analysis. Yeah, all right. For the statistical analysis, we are able to analyze the variances in data using the two-way ANOVA, using graph prism. And likewise, we're able to draw the dose response curve, and the EC50 was obtained using the excelligent integrated software. Now we got our result. This result is for the antioxidant content and the antioxidant activity. On the left-hand side, we can see the content, which is the polyphenol and the flavonoid, while at the right-hand side, we can see the antioxidant activity, which is the DPPH and the FRAP. We can also see from this table that there is significant differences in the result of the ethanol extract and the water extract from both sides, except for the FRAP, which seems to not have a statistical difference between the water extract and the ethanol extract. But generally looking at the result, we can see that the ethanol extract seems to be more active and has more content compared to the water extract in this regard. So moving forward, we did all of the analysis, which is for the lava cider activity. Here's the result for the lava cider activity. As I said earlier, that we did a 2-point analysis, concentration analysis, which is at 10 microgram per mil and 20 microgram per mil. So we are able to get a contrary result compared to the first result that we got in figure 1. So we realized that the water extract in this regard is more active compared to the ethanol extract. The water extract at 20 milligrams per mil is almost equal to that of the positive standard that we use, the chloroquine that we use. Then the cytotoxic analysis, here is a curve that shows the pattern, the relationship or the response of this cell to the cancerous cell. Because we use the HP2 cell, which is the human liver cell line. In this human liver cell line, A represents the water extract and B represents the ethanol extract. Mainly looking at the result, we can see that at A, the cell line continued to grow even though the extract were introduced. But at B, you can see the slump as the concentration at 1,000 microgram per mil and 500 microgram per mil, which shows cytotoxicity. Here is the result for the Vero, which is a non-cancerous cell, the African monkey cell line. So in this Vero, we can also see that the similar pattern is repeated here, where the ethanol extract is more active, starting from the concentration of 1,000 microgram per mil up onto 31.25 microgram per mil is cytotoxic. So this table summarizes the EC50. If you look at the table properly, you can see that under the EC50 of the ethanol extract is 0.04, and that is for the HP2 cell, while the ethanol extract for the Ferro is 0.02, which shows that the ethanol extract is cytotoxic, unlike the water extract. Now, further analysis that was done in order to quantify or to check the phytochemicals in this extract, we use the HPLC both at the positive and the negative ESI. So if you check proper, if you look properly, you can see that our base chromatograph shows that there are few compounds in the water extract, because A1 represents the water extract, while A2 represents the ethanol extract. And likewise, you can see that the ethanol extract has more peaks. And along the line, we're able to identify 54 compounds for the water extract. We're also able to identify 101 compounds for the ethanol extract. This is the result for the LCMS. So the LCMS also shows similar pattern. If you looking at the peaks, we can see that A1 represents the water extract, while A2 represents the ethanol extract. So it was done at both positive and negative ESI. And if you check properly, you see that there are more peaks on both positive and negative ESI for the ethanol extract. Now we came up with 140 compounds altogether, of which some of the compound overlap in both sides. So we're able to bring this table. The table is quite a long one, but I won't take you through all the table, but instead I will simply explain a little bit about what we have here. So the MF represents the molecular formula, the UV of the compound annotated, the MZ molecular ion, both positive and negative, as you can see in the column, and the MS product ion. Likewise, the identified compound, while A1 represents ethanol, water extract, A2 ethanol extract. So, but then I decided, for the purpose of this conference, I decided to pull up a pie chart to show exactly what is happening on this table by classifying the compound into 5 groups. So let's pay attention to the phenolic compound here. Likewise, the flavonoid here. So you can see that for the phenolic compound, we have 52% of the phenolic acid that we are able to identify in the water extract of 54 identified compound, while the flavonoid has 6% of flavonoids. 6% of the extract entirely. So looking at the next one, you see that there is a different trend here, where we have the phenolic acid, 2%. This is the ethanol extract actually, and the flavonoid is 33%. So the flavonoid here seems to be about 5 times or 6 times more than what we have in the water extract. Then we begin to imagine, could it be the reason why we have differences in the bioactivity of both extracts? If you check from this comparative table, you can see that the phenolic acid, the coumarin and furunocoumarin, and the acid and other hydroxy compound, they are kind of having a closer percentage in this table. But for the flavonoid, you can see that there is a huge gap between the two, and we begin to suspect that the flavonoid could be the reason why the bioactivity of both compounds varies from each other. Now, in my discussion, the flavonoid and phenolic compound has 33% and 42% in 70% ethanol extract respectively, and we believe that high flavonoid could have caused higher antioxidant activity and cytotoxicity effect recorded in the ethanol extract. And also breaking down the phenolic compound that we were able to identify, although I didn't really put it on this slide because of time and the constraint of slide. So we realized that hydrocinamic acid is a major part of the phenolic compound in the water extract, which was reported to have a strong antiemetic activities. And we believe or suspect that this might actually be the reason why the water extract is more active in the larvacidal assay. Now, generally, a flavonoid and phenolic compound are attributed to antioxidant, anti-inflammatory, anti-mutagenic, and larvacidal property of plant. So this plant has these things. Sorry?
Your time is almost up. You have about 2 minutes left.
All right, I'll manage that. Thank you. All right. So the ethanol extract are more active than water extract in most bioassays has been reported. Similar trend is recorded in this study, as I explained earlier. So in conclusion, the report presents the first comprehensive secondary metabolite profile that aim to provide chemical-based evidence for the health benefit of breeding Pilosa. So we use the HPLC and Quitov mass analyzer to analyze about 140 phytochemicals, which are predominantly phenolic acid, flavonoid, and fatty acid. So the plant extract shows that they are cytotoxic but not poisonous, because we do not identify any poisonous compounds such as rotenol, And this finding provides further evidence for the value of the plant as a source of functional phytoconstituent and explain the impact of solvent extraction. So here are the references that I used throughout this slide. And thank you for listening and your time. And apologies for the slide earlier. Thank you.
Oladayo. We don't have time for questions, but thank you so much for the interesting presentation. Without much further ado, we'll move on to the next presenter. And should we have time towards the end, we can always go back to ask Oladayo. Thank you so much. Our next presenter is Nompendulo Koza. You can go ahead and share your slides.
Okay, thank you.
And to avoid repetition, I'll let you tell us what your title is.
Good day. My name is Nompendulo, an ethnobotanical study of indigenous knowledge and uses of medicinal plants by traditional healers in the rural communities of Nkomazi local municipality in the Mpumalanga province. For centuries, Africans have depended on traditional medicine. Traditional healers are the traditional medicine knowledge holders and play a crucial role in African communities. They are found in nearly every village and are the first health practitioners to be consulted on both physiological illness and spiritual issues within these communities. Around 70 to 80% of South African Black population uses traditional medicine. Sorry about that. Indigenous knowledge on the use of medicinal plants is already passed down from generation to generation. The chain of if no one in the family is interested. Indigenous knowledge on the use of medicinal plants is at risk of being lost because most of the eligible youth to practice as traditional healers are not interested in learning about the practice. The traditional way of life in many South African villages is rapidly transforming because of economic development, Western education, urbanization, and improved access to healthcare and climate. The available research on the use of traditional medicine by the rural communities of Mpumalanga and under-researched. It only encompasses the council, which falls under the Bushbuckridge local municipality. And a detailed account of medicinal plants and associated indigenous knowledge for the Nkomazi local municipality is not available. Therefore, a thorough investigation and documentation of the medicinal plants usage and use pattern by the community of Nkomazi is necessary to preserve the indigenous knowledge shared by the locals. The study aimed to investigate aspects related to the collection and utilization of medicinal plants by traditional healers at Nkomazi local municipality. The objective of the study was to document medicinal plants collected and utilized by traditional healers, document medicinal plants used in treating COVID-19, to determine the most harvested plant parts, harvesting methods, and storage and packaging processes, to assess whether there is adequate knowledge amongst healers regarding environmental management regulation relating to the harvesting and sustainability of indigenous medicinal plants in Nkomazi. The area is located within the eastern parts of the Ehlazeni administrative district of Mpumalanga. The municipality has 8 traditional authorities and 43 villages. The area is dominated by Shangaan and Swati ethnic groups due to its proximity to Eswatini and Mozambique. The study adopted a qualitative approach. Individual semi-structured interviews were conducted with 10 traditional healers from 8 villages around Nkomazi local municipality. The semi-structured interviews were supplemented by observation, fieldwork, as well as extensive literature review. The questionnaire were divided into 4 sections: the biogeographical information on the participants, plants collected and used by traditional healers, conservation and legislation, and COVID-19 treatment. Traditional healers were sampled using the snowball sampling method, where few individuals who are knowledgeable about the focused topic are identified and asked to assist the researcher by referring other individuals relevant to the study. Analysis entailed transcribing the data gathered, analyzing all the participants. Was obtained from each individual who participated in the study. The traditional healers interview were a diverse group from a wide range, which included males, females, no formal to formal education, herbalists, and diviners. 60% of the interviews were Swati speaking compared to 40% being Tsonga speaking. The ages of respondents interviewed ranged from 24 to 60 years old, and majority of them were female. The results from this study are in accordance with a study conducted by Williams in 2004, who reported that 70% of the Faraday multi-market traders in Johannesburg were women. A total of 140 medicinal plant species covering 77 genera and 64 families were recorded. The species comprise different forms such as trees, shrubs, climbers, bulbs, and herbs. Table 2 represents the ethnobotanically inventory with detailed information such as the local name, family names, parts used, preparation method, medicinal plant uses. Out of the 140 species recorded, only 6 were used for this presentation. For instance, you can see that roots, bark, leaves are used to make a decoction that is taken orally to treat headaches, sinuses, colds, and diarrhea. The most common methods of preparation used by traditional healers interviewed included infusions, inhalations, ointment, and lotions. The most common methods of administration are orally, topically, steaming, and bathing. According to traditional healers, preparation and administration methods are dependent on the ailments being treated. For instance, Capucines tomatosa roots are boiled to make a decoction or infusion for the treatment of sexually transmitted infections, ulcers, and labor pains. Vepris reflexa roots are prepared by steaming for the treatment of flu or fever-related COVID-19 symptoms. About 30% of the traditional healers interviewed indicated that they consulted or treated patients with COVID-19. And they also mentioned that they use plants such as Vepris reflexa, Vinemia adonisus, Artemisia afra. However, there was some reluctance to share COVID-19. One of the participants indicated that scientists are stealing from them and they're not being knowledge. He said he preferred not to share the medicinal plants with me because research of stealing information from them and using it for their own benefit. And he's scared that whatever he uses might be commercialized and he will not benefit anything from it. The most harvested parts were the roots, which accounted for 45%, followed bark, leaves, and the whole plant. According to the study by Williams in 2007, where he found that the bark was the most popular plant part harvested for medicinal use in South Africa. However, in the study, it is not the case in the Nkomazi local municipality, where the roots are the most harvested plant parts. This preference for roots by the majority of traditional healers can be ascribed to the belief that the roots are the most powerful plant parts. Traditionally, healers use different objects for harvesting medicinal plants, such as spades and axes. You can see in the images on the right. Traditional healers also indicated that they practice sustainable harvesting methods. In a case where the whole plant is harvested, not every plant is harvested. In the case of bark harvesting, ring barking is always avoided. The wound is covered with soil to fight. The same applies to roots. Are left behind for the plant to survive. After harvesting the root, the soil is returned into the hole to cover the remaining roots for the plant to grow again. Traditional healers use various materials for packaging, including newspapers, plastic, and bottles. According to the traditional healers, medicinal plants can be stored for a long time with healing properties. Before storage of the collected medicinal plants are normally dried in the sun and then ground and kept in plastic bottles. 90% of the questioned healers did not have any knowledge of the various national or provincial environmental laws relating to medicinal plants or about rare data species. The traditional healers were also unaware of any protection. The finding that 90% of the traditional healers did not have any knowledge of environmental legislation means that the majority of Nkomazi traditional medicine community is probably operating outside the framework of national and environmental legislation regarding the collection of medicinal plant species. Indigenous knowledge of medicinal plants at Nkomazi local municipality is characterized by a rich diversity shown by 150 species and have been recorded. Cultural conservation practices are still in place in this community. However, there's still a need to educate community members about plants. Further research on ecological and cultural conservation efforts are needed for the sustainable use of medicinal plants. The findings presented in the study present a significant contribution to the South African ethnobotany and the cultural heritage of South Africa. Thank you.
For this lovely presentation and interesting information that you've given us. I open the floor to questions. I think the first question that I see on the chat is directed to Oladaye, but this second one, it says, are the traditional healers aware of the IKS Act 6 of 2019 and what it means for them? This comes from Prof. Aremo.
I do not think, I think few of them might know about the Act, but the majority of them do not know about the Act, because when I interviewed them about protected species and what it means and the other national structures, they did not have any idea of what they meant. So I don't think majority of them would know or are aware of it.
Can I just ask, are they registered with any traditional healers organization?
Yes, they are registered, but for the sake of this presentation, I had to cut down few sections because it was going to be over time. So yes, 90% of them are registered, but 10% of them said they're not registered because some of them are working with their mothers and the mother is registered.
Okay. All right. Thank you so much.
Thank you.
Um, thank you very much. I think for our next presenter, we have time for our next presenter. Thank you, Nompendulo. Our next presenter is Edward Konona. Kunwana, sorry, Edward Tato Kunwana. Are you ready, Edward? Um, Perhaps he's waiting for the time to lapse because our previous speaker ended pretty early. Um,
Edward?
if Olayade is still around, Idris, would you like to answer the questions in the question and answer chat? Are you still available, Idris?
Consumed as food. Basically, the part of the plant that is consumed as food is the leaf of the plant. It is being used as spices, particularly in Sri Lanka, where this plant is being dried and used as spice because of have this aroma. So, but the second question is, did you compare the antioxidant activity of the plant extract with reference compound? Yes, we did. We compared it with routine, but because we are focusing on the differences of between the ethanol extract as well as the water extract, so we decided to leave the reference compound because we know that there are information about the reference compound there everywhere in other literature. So we decided to concentrate on the two extracts. So I hope I answered the question to the satisfaction of the
Okay. Thank you so much, Oladayo.
all right, thank you, ma'am.
We now have Edward on the stage. I will let Edward present his title. He's from University of Pretoria. Welcome, Edward, and good luck with your presentation.
Okay, thank you very much.
Yes, we can hear you.
My studies on variation in biological activity of Thyroxamol obliquum collected in 4 different geographic regions of South Africa. I'm having challenges now. I don't know what's happening. Oh, it's gone.
Hi, Edward.
My
Edward,
slides
can you
seems
hear
to have
me?
disappeared.
Yeah, let's try and share it again.
That I send them on the drop because it seems like mine keeps on disappearing.
Yeah, I don't have access to the Dropbox before.
Okay,
Just open your presentation because you had it. So You had it, so
Yeah.
let's just open your presentation. Do you have it open in full screen?
Yeah, I'm there, almost there.
Okay. And when you advance your slides, you must
Still loading.
it's fine. Edward.
Yes, I'm with you. I'm still trying to load it. Still loading. I don't know what's happening. Seems to be a bit slow.
When you say loading, what is loading?
The PowerPoint app itself to open up the presentation.
Oh, the PowerPoint itself.
Yes.
Okay. Edward, do you want to send me your presentation? And then, Ida, are you there? Perhaps we can go on to Pca while I get Edward's presentation from him.
Yes, I think that's the best thing to do.
Okay, Edward, you can email it through to SAAB Tech Support
Okay.
at xlmconferences.co.za. I'll put it in the chat.
Okay.
Okay.
Okay. Thank you, Edward. Kiabetu, are you available? Are you ready to present? Kiabetu Masike? Kiabetu? Um, Colleagues, let us see if our next participant is the next participant is available. Mabuza. But
The boosters are online. We go to JK.
sorry,
We can go to JK Madisha.
okay. J.K. Madisha.
Jacobus.
Jacobus. Corey Madisha, are you available?
Okay, so it seems we're back at Edward. I don't know if perhaps Edward can speak without his slides.
Hello.
Yes, Corin.
There we
Hello,
go.
dear.
Then my
Please go ahead.
This
No, it's not.
Yet.
okay. Slide. You hear me? Hello.
Yes, we can hear you.
Hear me?
Yes, we can hear you.
Right.
Can you share your presentation for us?
Yep.
No, it's not.
I'm trying to.
Okay, can you open your presentation? Can you open your presentation in Slideshow?
Okay, let me put the camera.
Sorry. Is your PowerPoint open?
I've opened it.
And you've put into Slideshow. So
It's
your
on my
whole
screen.
screen.
Yes, it's on my screen now.
So your whole screen.
Are you able to see it? My
Sorry.
PowerPoint on the screen.
No.
Yeah.
Yeah. Just hold. I'm going to talk you through it. So your whole screen is open. Is this just the PowerPoint, correct? The Slideshow. You can't see us or anything else. Is that correct?
Yes.
Okay, so your whole screen is the Slideshow. Are you on a Windows or a Mac laptop? Are you? Yeah, are you on a Windows or a Mac laptop?
Right. It's a Mac laptop.
It's a Mac. Okay, do you have an F3 key?
Yes.
Okay, press the F3.
I don't.
You will then be
see
able
it
to
on-
see Google Chrome.
Okay. I don't.
Do you
happen
not
to
have an
see-
F3 key?
No, F3 key on my okay. On the
your
laptop.
keyboard?
All right.
Yeah. Okay, so now if you press the F3 key, you'll be able to see all your open applications, and you can see Google Chrome.
I'm not seeing it. Display the settings, slideshow, and show the taskbar. That's what it's showing me. It says display the setting. Sub slide. That's what it's showing me. Did I go
Okay,
to show the taskbar?
on your keyboard, at the top of your keyboard, do you?
Yes.
It's about the third button from the left-hand side. It's it looks like three squares. So it's two squares, rectangles above one another, and then next to it there's a portrait rectangle. Do you have that on your keyboard?
I have a pen and then now goes to the show. Are you able to see my slide on the screen?
No, you won't. It won't be sharing it because you haven't selected share. I'm trying to get you back into Google Chrome.
Okay, so I should what end my slide?
No. You must keep the slides open. You need to get back to your desktop.
Okay.
So you either need to press F3, which will get you back to your desktop, and you'll be able to see the other applications, and you can choose Google Chrome to come back to Chrome, back to Remo. Key on your Mac, then it's three buttons, it's three lines. It's usually the third one from the left. It's got two rectangular lines, one above each other, and then one to the right of those. So it's all on one button. It's about the third one.
F3, it's written
So,
F3.
yes,
F3, I
F3 on
press
your
it, but
keyboard.
nothing is happening.
I'm not too sure. Have you sent through a backup of your presentation?
Yes,
Where did you send it to?
to the Dropbox.
Okay, can you email it through to me as well, please? Email address in the chat.
The email address in the
Okay.
chat.
Yeah, and then we'll move
Okay.
on to our next speaker, because I see Pca Biplot is online. So if you can email me your backup presentation, in the meantime, and we'll come back to you. Okay,
It's on the chat.
it's on the chat.
Yeah.
It's on the chat. My email address, correct?
It's
Okay.
written
SAAB tech support at xlmconferences.co.za. Okay, we'll come back to you as soon as I've received it. Okay, thank you.
Okay, thanks.
I think Pca Biplot is ready to share. Pca Biplot, are you ready to share your presentation?
Hi,
Hi, Pia.
how are you?
You can
Yes, I am. I can hear you.
please share.
Okay.
Pca Biplot is going to present on the phenolic characterization of Protea pure and hybrid cultivars by liquid chromatography, ion mobility high resolution mass spectrometry. That's LCIMHRMS. When you're ready, Pca Biplot.
Morning. I'm worried that the internet might not hold. Is it okay if I leave the video off? Can you hear me? Hello?
We can hear you. We hear you.
Oh, okay. Thank you. I can continue?
Yes, please continue.
All right. Thank you so much. As that is the title of my presentation. Now, the Proteaceae family is endemic to the Mediterranean type regions, such as parts of South Africa and Australia. Within this family, there are different species, about 1,700 species to be specific. And within these species, we have the Protea species, and the Protea species are located in fire-prone and semi-desert environments and pollinated by sugarbirds, sunbirds, and rodents. The Proteas exist as both species and cultivars, and because of their very beautiful flower heads, about 3 million were exported from South Africa in 2017. Historically, Protea plant tissue has been used for a range of applications in leather tanning, the production of wagon wheels, and the nectar for the production of cough syrup. Different studies have been conducted on the proteas, such as population genetic studies, transcriptomics. However, studies dedicated to the phytochemical composition of protea are limited, and this has resulted in few phenolic compounds and polyol sugars been identified as lead constituents. Despite the very vibrant flower heads of the proteas, they are prone to a post-harvest disorder referred to as leaf blackening. Leaf blackening can occur at any point in the post-harvest chain, and this often results in consignment rejection by the importers, as well as this is a loss for the farmers. Now, leaf blackening can present itself, how it shows itself is that it's the formation of brown and black discoloration on the leaves and manifests in different ways, such as the leaf tip, the leaf base, the midrib area, as well as the lateral leaf margins. It's not fully understood what is causing the leaf blackening or the processes behind leaf blackening. The reduction in the carbohydrate content has been suggested. Enzymatic and non-enzymatic processes have also been linked to leaf blackening, but the process is still not fully understood. And I think this could be due to the fact that some species and cultivars are susceptible to blackening, whereas some are not prone to leaf blackening. This has called for more studies to be conducted, possibly at a metabolite level. Now, metabolomics as a study is, it looks at, it provides insight into an organism's metabolome at physiological as well as pathological states, where targeted and untargeted metabolomics approaches can be used to identify trait-specific markers, and this can help improve commercially important traits. The analytical platforms that can be used are LC or LCMS, NMR, and these platforms can then be used to identify these trait-specific metabolites. This brings me to the aims of the study, which is to evaluate the phenolic composition of Protea species and cultivars, whereby we want to firstly characterize the phenolic compounds in the Protea species using UPLC PDA ion mobility high-res mass spectrometry, and then identify the possible metabolites or phenolics that are linked to leaf blackening in Protea species and cultivars with the aid of metabolomic tools. I'm going to provide a brief overview of how the experiments were conducted for extraction plant tissue samples, and by that I refer to the leaf tissue as well as the bracts tissue, were extracted with 50% acidified methanol. 37 plants were available to us, which were separated as species and cultivars from different farms. The extract was then sonicated, centrifuged, and the resulting supernatant was analyzed using UPLC ion mobility MS, specifically a UPLC SYNAP instrument. The resulting data was then uploaded onto different softwares, where XCMS, which is an online software, was used to process the data. The resulting data matrix was then uploaded onto MetaboAnalyst. MetaboAnalyst provides you with different figures, such as a principal component analysis, which is a PCA. And then for the ion mobility data, we use DriftScope to facilitate in data analysis. Once these compounds, or rather to identify these compounds, we use different databases, and those compounds were then annotated in a situation whereby we had a standard or NMR was used. We were then able to identify these compounds. Now for the results, this is a UPLC MS chromatogram, rather UPLC chromatograms, which represents the two species, Cynorhodis and Merifolia, as well as the cultivars Limelight and Black Beauty. What the numbers represent is how we numbered the different peaks, and this will make sense in the next slide. And what you see on the x-axis is the retention time. The retention time just tells you how long it took for the particular peak or compound to elute from the column. As you can see from these four chromatograms, there are chromatographic differences, suggesting phenolic compound differences between the Protea plants studied. Now, this is the slide that I was referring to, whereby the numbers on the chromatograms are reflected here. And each peak represents a compound, and each peak, if possible, was annotated or identified. And the retention time is shown in that column. And then the mass to charge represents the molecular weight of the particular compound. So because the compound has to be charged within the instrument, then it has a mass to charge ratio, which is just the molecular weight minus a proton. Then that particular precursor is exposed to a collision gas, and that collision gas provides us with fragments, and we use these fragments on the mass spectra to then identify the different compounds within the extract. The SYNAPT instrument is a high-resolution instrument, therefore we're able to calculate the chemical formula, which then aids us in identifying the compounds. Then the CCS value is not particularly important, but what is important about the collision cross-section is that the mass spectrometer, if you're dealing with isomers, in some cases, it's not able to differentiate the isomers. For instance, for compound 2 and compound 3, they have the same precursor, they have the same fragments, which then makes it difficult to identify these two compounds. But if we use mass spectrometry, we're able to calculate the collision cross-section, and we can then identify different compounds or different isomers based on their collision cross-section. And therefore, these two compounds was beta and alpha arbutin. Then within the 37 plants that were studied, we saw that there was a compound that was quite prominent in all the 37 species as well as cultivars that were studied. We couldn't find or identify this compound in literature, so we called it an unknown compound, but this is how we have it. It has its UV-vis chromatogram or its absorbance, and then its extracted ion chromatogram at mass to charge 325. We have its ion mobility profile as well as its mass MS spectra. So to identify this compound, we then use NMR studies, which is 1D and 2D NMR, which helped us to identify this compound as kefir polygallitol. The polygallitol sugar is quite interesting because in a study involving Australia, New Zealand, as well as South Africa Pca family, from the 82 species that were studied, only the species that were endemic to South Africa only contained this polygallitol sugar, suggesting some chemotaxonomic marker or potential of this particular compound. Now, initially, I spoke about the post-harvest disorder leaf blackening. So what we did in this study is to look at all the different species that were available to us and try to see which ones would have different compounds based on their susceptibility to leaf blackening. And what we found was we have a Pca Source Plot. Now, Pca Source Plot shows you differences and similarities between samples in this case, and species in this case, rather. And from the Pca, we see that we have 3 different groupings. And these 3 different groupings can be linked to the physical susceptibility of these different species to leaf blackening, whereas group 1 and group 2 are oftentimes the species that are susceptible to leaf blackening, whereas group 3 is your species that are not susceptible to leaf blackening. And we wanted to see which metabolites could be linked to group 1 and group 2, as well as group 3, so meaning which particular compounds are linked to the susceptibility of the Protea species to leaf blackening, whereas the others, which are in group 3, are not susceptible to leaf blackening. Figure B or 7B is a hierarchical cluster analysis, which just reflects what we see on the Pca Source Plot, which arranges the different samples based on how similar or different they are. So now we wanted to dive into these particular metabolites that can be linked to leaf blackening. So this is a Pca Biplot. So the Pca Biplot shows you the features. In metabolomics, we refer to the mass to charge and retention time as a feature, but it's just a peak. And the peaks or the compounds or the features that are linked to group 1 are as such. And the same thing, the features that are linked to group 2 were identified, as well as the features that are linked to group 3 were identified to show us which particular compounds are linked to the non-susceptibility of these proteas to leaf blackening. Then we did a box plot to sort of show you how these compounds are represented across the different groups. So an example for group 1, we have the compound hydroxyquinol, which is actually a hydroxyquinol derivative, which is this group right here. And then for group 2, which is arbutin, I spoke about it earlier, which is a hydroquinone derivative. And then group 3 has these compounds. These tend to have these catechol groups. These catechol groups are quite interesting because they are known to be antioxidants, whereas these hydroquinone and hydroxyquinol groupings are compounds that tend to oxidize. And when they undergo these oxidation reactions, they produce very black products, which will also suggest that maybe this is what is causing the leaf blackening in this Protea species, where the species that are susceptible will have these compounds, and the species in group 3 that are not susceptible to leaf blackening will have these antioxidant properties. Then I did a time study whereby I took a cultivar from group 1 and group 2, which is referred to as carnival. And over a 3-week period, I collected the leaves and analyzed them after I'd collected all these leaves. I went through the same process as I showed you in the methods section. And then I uploaded the data onto Metabolomics to sort of get a metabolome view of what is happening throughout the blackening process over a 3-week period. And the y-axis, we see the pathway enrichment analysis. On the x-axis, it's the pathway topology analysis. So a minus log p value greater than 2.9, which is like 3, represents pathways that are affected, and and an impact value greater than 0.1, which is these 11 and 12, represents pathways that are impacted. So the pathways that are affected were linked to sugar metabolism, and the pathways that were impacted were linked to the phenylpropanoid pathway biosynthesis, flavonol, and the flavonol biosynthesis, which is linked to the phenolic and the flavonoid pathways. Now, the affected pathways, which is the sugar pathway, which is the pathways linked to sugar metabolism, are interesting because initially in the introduction, I had mentioned that leaf blackening is associated with the reduction of the carbohydrate content, specifically sucrose. In some studies, it has been found. So what we are seeing here sort of linked to what we have, what previous information or previous data suggests. The darker the red, or the darker the color, it represents how affected a pathway is, and the bigger the circle tells you how impacted a pathway is. Now, to conclude, the study reports the first detailed phenolic or phytochemical investigation of Protea species and cultivars. We report the presence of 166 compounds, which are made of 4 organic acids, 3 sugars, 61 phenolic acid esters, and 25 flavonoids. And there were some compounds that were unknown. They were prominent, but they were unknown and we couldn't identify them. And then a new hydroxycinamic acid derivative was identified using NMR studies. Using untargeted metabolomics tools, we're able to investigate differences in metabolite levels among Protea species and cultivars. And these studies, or this untargeted approach, points to the levels of benzene trials, in some cases anthocyanins, hydroquinone derivatives that could be linked to the susceptibility of Proteas to leaf blackening. Whereas in group 3, organic acids and these compounds that have catechol groups are linked to Protea species not prone to blackening. I'd like to acknowledge the people listed here and the farms that were gracious enough to give us the plants. Thank you. I will take your questions.
We have about 3 minutes for questions. We welcome questions from the floor. Well, there are no questions in the question and answer box. Okay, there goes one. There goes one. Two. The first one is, how easy is it to scientifically identify the species used? Oh no, I'm sorry. I'm asking the wrong person. Sorry. What is the significance of the findings to the different stakeholders? For example, farmers.
Yes, the understanding is once we understand or know which metabolites are linked to leaf blackening, then we can then have the next generation of cultivars where we choose to not have these particular compounds, or we silence certain genes that may be linked to the production of these compounds. And as such, the fact that some of the consignment, when some of these plants, when they're exported, then they are returned, we then stop that process, which is a loss for the farmers. I hope I answered that correctly. I hope I made sense.
Yes, thank you. The other comments from Prof. Generic. Well done, Keveswe. Is there any strategy to improve resistance to blackening by breeding?
At the moment, what the farmers do is add, during the harvest process, they add the stems in sugar water. So, which then links to the fact that maybe the sugar metabolism is affected during the leaf blackening process. So they'll add the stems in sugar water and keep them in there so that once the flower head starts opening up, it does not affect the sugar metabolism. So we sort of add sugar for the time being. But it's a problem because it's difficult to export these plants when they're in sugar water. So if we can silence certain genes, it will be helpful moving forward.
Yes, thank you, Keveswe. We are now out of time. And I think we are ready to welcome back Mabuza. That is MJ Mabuza. Are you ready? Baby.
Hi, am I audible?
Yes, we can hear you.
All right, I'm trying to project the presentation, but it's not appearing on the window. I don't know why.
You sent your presentation to.
Hi.
I uploaded it on the
Do
Dropbox.
you have your presentation open in Slideshow?
I have it on the Dropbox, yes.
No. Do you have your presentation open in front of you in
Yes,
Slideshow?
it's
Yeah, you can. So you only see your presentation. You see nothing else.
yes.
Are you on a Windows or a Mac laptop? On the Mac, okay. You have it. Sorry. Hi.
Tabitha, do you have two devices on the platform? Perhaps you can shut off one. If you have two devices on the platform, perhaps you can shut off one.
There's only one that I'm using.
Okay, now we can hear you clearer.
Yes, I'm on the presentation.
Okay, So you're on the presentation, and have you pressed F3? Do you have an F3 key?
Yes, I've pressed F3.
You've pressed F3.
Huh?
Okay, now select to come back to Google Chrome.
That's it.
Open Google Chrome.
There's still nothing appearing.
Are you in Google Chrome?
Yes, I'm on Google Chrome.
Okay, so you've come back into Remo.
Yes, I've come back and I've selected
Okay, then
the option for window.
then click share.
There is no presentation to share that's appearing here.
Under window,
Yes,
there's nothing there.
there's nothing there. And this was the
Okay,
same mistake. This was the same
Okay.
thing.
Have you checked your security settings?
No,
You need
I'm not
to,
aware of that.
if you're on a Mac, you need to allow screen recording.
Screen
So
recording.
you need to go into your settings and you need to allow screen recordings.
Okay, settings.
Okay, so I think while you are doing that, I think we should move maybe back to Edward, Ida.
Yes.
Ida, are you there?
Indeed. Edward, are you ready?
Edward, are you there?
Edward? Or
Jacobus,
maybe
are you there? Is Jacobus there? There's Edward.
yes.
Okay, Edward, you there?
Yes, I'm
Welcome
here.
back, Edward.
Thank you.
Okay, Edward, do you want to try sharing your screen again, or do you want me just to do it for you?
I think you can just do it for me just to save time because I think it's giving me problems on my side.
Okay, you just need to let me know when I need to proceed to the next slide, okay?
Okay.
Okay.
Okay, my presentation is on. Am I good to go? Okay, my study is on variation in biological activity of Pterozomal obliquum collected in 4 different geographic regions of Southern Africa. We can move on to the next point. Okay, medicinal plants are known to vary in quality and consistency of active secondary metabolites. This may be as a result of ecological and environmental differences. Secondary metabolites composition of medicinal plants are known to be affected by a number of environmental factors such as altitude, soil type, geographic location, and change of season. The change in temperature associated with climate change are causing noticeable effects on the life cycle, distribution, and phytochemical composition of the medicinal plants. All these factors contribute towards in the variation of the pharmacological activities of the medicinal plants across various regions. We can move on. Thyroxamol obliquum is used traditionally to treat various infectious diseases, including fungal infections, tuberculosis, inflammatory diseases, and also for ethnoveterinary purposes. We can move on. The plant is mainly found in the eastern coast of Southern Africa in countries such as South Africa, Mozambique, and Zimbabwe. We can move on. Our aim is to investigate the variation in biological activity of Thyroxamol obliquum extract collected in different regions of South Africa. In order to achieve our aims, the following objectives were set. The first one was to collect the leaves in 4 geographic areas in the same period or season. Our extraction and fractionation of the leaves was done using solvents of varying polarity. In terms of our biological activity, we managed to analyze the antifungal, antioxidant, and the cytotoxic activity. Next one. We can move on. Okay. Our leaves are collected at Nelspruit at the Lowveld Botanical Garden, and at Pretoria at Sunby, and also University of Pretoria main campus, and lastly at Johannesburg Walter Sisulu Botanical Gardens. After collection, our leaves were dried and grinded into powder, and 5 preliminary solvents were used for extraction, and acetone leaf extract was selected for fractionation. Unfortunately, due to the unavailability of enough plant material, only extracts from Nelspruit and Pretoria Sunby were able to be fractionated, and solvent-solvent fractionation was used. We can move on. To determine our antifungal activity, microplate cereal dilution was used, and 3 fungal strains, including Candida albicans, Cryptococcus neoformans, and Aspergillus fumigatus were used. In terms of our cytotoxicity, the MTT assay was used, and the extract were tested against Vero monkey kidney cells, breast cancer cells, hepatocarcinoma liver cells, and adenocarcinoma lung cells. For our antioxidant activity, we used the DPPH and ABTS assays. We can move on. Now, coming to our antifungal results for crude extract, we've noticed that Walter Sisulu and Nelspruit were the most active regions against all 3 pathogens. We've also noticed that acetone extract had better activity compared to the other preliminary solvents that were used, and we've seen that Nelspruit acetone had better antifungal activity compared to all other regions with MICs of 0.5 to 0.8. We can move on. When coming to our fractions, we've noticed that chloroform fractions had better activity across board against all 3 fungal pathogens. And we also noticed that Nelspruit had better activity for Aspergillus fumigatus and Cryptococcus neoformans with MICs from 0.6 to 0.8. However, for Candida albicans with MICs of 0.63. We can move on. Coming to our cytotoxicity, we noticed that acetone extracts were moderately toxic with IC50 values of 0.6 to 0.9, except for Sunby, which was a bit more toxic with IC50 of 0.075. We've also noticed the same trend for Sunby fractions, with both chloroform and hexane fractions having IC50 values of 0.075, while Nelspruit fractions had moderate toxicity with IC50s of 0.07 and 0.19. Water extracts were generally non-toxic. However, we noticed that cold water extract had higher IC50 values, while hot water extract had low IC50 values. The selectivity index indicated the presence of cytotoxic effect. We can move on. Moving to our cancer cell lines, we've noticed that acetone extracts from Sunby were moderate against breast and liver cancer cell lines with IC50 values of 0.04 to 0.09, and University of Pretoria extracts were toxic to liver cancer cell lines with IC50 of 0.076, and also moderately toxic to lung cancer cell lines. Although Nelspruit acetone extract were not, Although Nelspruit acetone extracts were not toxic, however, the chloroform fractions were toxic with IC50 of 0.01. We can move. Coming to our antioxidant activity, we've noticed that all water extract had better activity compared to acetone extract and fractions. When looking at our hot water extract, we've noticed that Sunbe had better activity compared to Nelspruit. While looking at our cold water extract, we noticed that Nelspruit had better antioxidant activity compared to Sunbe for both DPPH and ABTS. We can move. Coming to our conclusion, we can safely conclude that antifungal activity varied with geographic location, as we've seen Nelspruit acetone extract having better antifungal activity. The next one. There were no major cytotoxic differences observed on acetone leaf extract based on geographic areas, except for Sunbe acetone extract, which was a bit toxic. Compounds responsible for cytotoxicity to Vero cells may largely be non-polar compounds. All water extracts were non-toxic against the tested cell line, and this is very good as traditionally water is used as the main extractant. So we've noticed that the water extracts were not really toxic against Vero cell lines, well, except for Hetfield, which was a bit toxic. We can move on. In terms of acetone extract, we've noticed that they had moderate toxicity against cancer cell line, except for Hetfield acetone leaf extract, which was toxic against Hep G cells. Next one. Same results were there for chloroform fraction, which was also toxic against Hep G cells with IC50 values of 10 microgram per ml. Water extract had the best free radical scavenging activity compared to acetone and fractions. Our future work includes the isolation of bioactive compounds, which we've already started on, also anti-inflammatory activity, and we are ready to do the thin layer chromatography and also UPLC-MS analysis. We'll also be evaluating geographic-based metabolism variation analysis. I'd like to thank my supervisor, Prof. Lindy, co-supervisor, Dr. Tanyani Ramadwa, and Prof. J.N. Ilof, and Dr. Sanan Kadimin. Thank you. I'll take any questions.
Thank you very much, Edward. We'll take a question or two. If there are any questions. In the absence of questions, we'll move on to Jacobus Madisha's presentation. Thank you so much, Edward.
Thank you.
Okay, I think I may have hurried, but you can see the questions in the chat. Thank you.
Yeah. Okay.
Are you ready, Jacobus? You
Hello?
can share your slides, or would you like Jackie to help you with your slides?
Straight from me. I don't have a problem anymore. You are able to hear me?
We can hear you.
Hello?
Oh, yeah. Okay.
Yeah.
Share the slide for me.
Okay, your slides are on now, so you can start.
Yeah. Thank you very much.
Good morning. My topic is antibioform, antioxidant, anti-inflammatory of selected temporary plants used in the cure of veterinary dermatological infections. My supervisor is
written there.
The next slide. Next slide.
You, Jacobus. We can hardly hear you.
Hello.
Can you go?
Hello. Next slide, please.
Next slide. Right.
Since it's commonly the antibiotics, which is caused by the biofilm activities. Biofilm are the ones that are together in communities which express the increase in the resistance.
Now, with the development of gene variation, then they ended up working against the antimicrobial in a search for newer product all the time as to overcome the resistance of the
and the resistance of bacteria as they grow. Veterinary dermatologists have created a lot of problems lost in terms of the stocks
or the stocks that does not look healthy, and also the one that is caused by the ticks that causes the livestock, which the livestock prices are affected, and farmers are working hard to control veterinary dermatological infections so that livestock remain healthy. Can you go to the next slide? Next slide. Right.
Plants are used in the form of herbal drugs to combat the
infections and in order to control the wounds and other that are related to the dermatological infections on the animals. The animals they are given that, or it is applied on their skin. As the skin, it's the biggest part of the body. So if it is, the animals becomes more uncomfortable. The
biofilm of the plants that are selected, and also the synergy
between the plants themselves. And we've also tested the synergy as we have used that, but we did not include it here for the study as there was time constraint. The study aimed at investigating or validating dermatological infections in Limpopo, in South Africa, and to assess their suitability in terms of being used by small-scale farmers, as small-scale farmers are economically disadvantaged. They cannot be able to purchase the also with the aim of either combining them with the synthetic drugs so that the resistance of the bacteria are
the next slide. Next slide. Results for the result interpretation 0.2
was considered as excellent and was considered as good activities, and anything above 1 was considered as moderate. And for the analysis of toxicity, the value 30 is considered moderate, and the below 30 is considered as non-toxic. And it might the comparison between the animal model and the model that we use in the laboratory, they are different. As when you go to the animal model, then the toxicity might differ in terms of the health of the animals. The next, please. That's how we'll interpret the results. The next slide. Can you go to the table of results? Hello? The table, that's conclusion. We'll go back a little bit. All right, here is the table of results. Please, can you stop on it? All right. Can you stop on it, please? Right. If we look at the plants on the table results, the plants shows that. Hello.
I can't stop on it. It keeps on skipping through. So I'm not so sure. Yeah.
Okay. All right. I've opened it. If you look at the plant A, that PA, which is P. aeruginosa, which it's 0.7, and also SA, it's 0.08, and also E. coli, it's 0.63. And also looking at their combination, now the MIC start to reduce, going down, meaning that they are more effective, or the MIC, when comparing them with gentamicin, you are able to see that in the combination, when you combine the two plants, then you are now able to find something more closer that we find in the synthetic drugs, even in terms of the MIC, and also the cytotoxicity of the plants. If you look at them, you are able to see that the cytotoxicity is very low, and also the DPPH also shows that it worth to be noted. But also when you go to the biofilm, the preformed biofilm, which is at the end of the slide, it's 57 for the first plant, and also the attachment it's 73. The plant shows that it can inhibit the biofilm, but when you combine the two, the percentage goes higher, meaning that it now can control the biofilm much better. When go to the second plant and the third plant, if you combine the two plants, then the more you increase the in terms of that, and also the biofilm also increase in terms of the numbers, meaning that the herbal combination, the one that are working, although we selected the herbal combinations that are common into they are common into the Barbary tribes from different areas where we selected the combination. And also the method that we have used, we have started with ethnoveterinary first, and then we survey, so that we collected different types of the combinations and the plants. We only chose 5 plants that are appearing several times in different Bapedi ethnics, and also which are used also in the surrounding town. And we decided to now report here only on the acetate extract, as we have also done the extract of others in terms of the combination, combining in the same plant, combining the extract of ethanol water, giving us also the results. But due to time constraint, we could not include this presentation. And also the MIC, we have used area of 1998 method, but we also used the different one. We used the biofilm strain in the MIC, so as to be able to compare the two strain in terms the biofilm and the one that is not biofilm compatible, in order to be able to see the resistance or the damage that the biofilm are able to cause. And then again, the limit vary in terms of the conditions, or how it is applied into the two systems. So those are some of the findings that we have find. And then after, we realized that the plants, to a certain extent, are able to manage the biofilm, especially enhance it more when they are combined. But over combining, it's also one to say it becomes no longer effective, or it increases the cytotoxicity. We limited the combination to 3 plants, and also we limited it also to 3 extractions when doing it from 1 plant. And in some cases, the reduction of the cytotoxicity, the combination of the plants, then this results substantiate the safety to some extent, and the efficacy of traditional use of the plants dermatological effects. Thank you.
Do we have any questions?
Any question? Hello?
Okay. There are no questions in the question and answer box, neither are they in the chat. So thank you so much, Giacobas, for your presentation. Thank you. We will now move on to our next presenter. Yaki, if you can, may you please share his slides for him? He has requested that you share the slides for him. Mcebisi Mabuza. Our next presenter is Mcebisi Mabuza.
I don't have the slides. Has it emailed them through to you? Hello.
Hi.
I haven't received your slides. Have you emailed it through?
I have uploaded on the Dropbox.
Can you please email it through?
All right. May I please have your email?
It's in the chat. It is
All right.
Can you see it there? SAAB tech support at XLM conferences.
I'm just trying to find it. All right, I got it. I'll send it through just now.
Okay. I think perhaps if he can start speaking without 10 to 12. Ida, are you there? Ida?
Mcebisi, would you be able to start without your slides? They'll catch up with you once Yaki gets them.
Yeah, I can. Let me just quickly send it just now. I can start without the slides.
All right.
Okay, as soon as
Because
I
we
receive
do
them,
have a few.
yeah, as soon as I receive it, I will I'll take it put it up for you.
All right, thank you. I've sent the email just now. All right, can I start now? Hello?
Can you hear me?
You can begin.
You can start, Mcebisi.
All right, thank you. For that, I'm going to share a presentation on the antiplasmodial properties of extracts from Pca capensis. So, malaria is a vector-borne disease which is caused by different Plasmodium species. About 5 different types of Plasmodium species, and the most deadliest one is Plasmodium falciparum. Malaria has a very complicated life cycle. Upon feeding in the human blood by the mosquito releases what we call sporozoites into the human host. And these sporozoites first colonize the liver, where they mature in the liver, forming what we call merozoites. And these merozoites in the human host then mature and colonize the red blood cells. Upon colonizing the red blood cells, they develop into schizons, and schizons mature and result, or rather, the schizons mature and cause the red blood cells to burst, releasing both male and female gametes of this parasite, which then continues the life cycle. When we look at the burden of malaria, there's about 50%, or rather more than 50% of the world's population is actually at risk of contracting malaria. And Sub-Saharan Africa records the highest burden in the world, and the burden is actually worsened by different socio-economic factors. Malaria is actually considered as a disease of poverty in Africa, and it's also believed to be a causative agent of poverty. The burden, especially in Sub-Saharan Africa, has not been completely eradicated. When you look at the global distribution map of malaria, you can see the bulk of the burden is actually found in between the tropics, where the temperatures are a bit warm enough to allow the parasites to proliferate in there. When you look at malaria in South Africa, it's endemic to 3 provinces, that is Limpopo, KZN, and Mpumalanga. And about 10% of South Africa's population is actually at risk of contracting malaria. There's about 2 transmitting vectors in South Africa, that is Anopheles arabiensis and Anopheles funestris. These are the 2 most important transmitting vectors transmitting malaria in South Africa. Malaria elimination it's possible to eliminate, especially in Southern Africa. This can be achieved if different countries can work hand in hand with each other, where malaria is mostly endemic. Countries like Zimbabwe, Zambia, and Mozambique, if they can come up with plans that can actually benefit all these neighboring countries, malaria can actually be blocked from being transmitted. When you look at the chemotherapeutics, drugs are the most common chemotherapeutics to treat malaria, and there are different types of drugs which actually target the Plasmodium at its different stages of its life cycle. 2 drugs that have been used earlier, or rather discovered to treat malaria, quinine and artemisinin. These drugs were used quite extensively in the past 20, 40 years. Despite a wide variety and availability of different drugs to treat malaria, the burden still has not been completely eradicated. Drug resistance, especially to mono and combination treatments of malaria, continues to be a problem. Now, to work around this problem, this problem necessitates the discovery of new drugs with diverse chemical structures and diverse mechanisms of action. And to solve this problem, plants have been identified to be the best sources for new drug development. This is because a large majority of prescribed drugs have actually been sourced from plants. Plant metabolites have not been extensively explored, and this sets the stage for a further study of these metabolites for the possible discovery of other vital constituents that can be used as active ingredients. The chemical structure diversity in plant metabolites warrants them to be the best sources of drug discoveries that have different mechanisms of actions and different chemical structures. The ethnomedicinal uses of Pca capensis include diarrhea, sexually transmitted infections, aphrodisiac, malaria, constipation, and ringworms. Indigenous people prepare these different plant parts, including fruits, the leaves, the stem barks. They are prepared differently, and they are administered in different ways to treat these different diseases. This brings me to the aim of my investigation. The aim of my research was to investigate the antiplasmodial activity of Pca capensis and to apply GCMS and metabolomics to dereplicate known antiplasmodial constituents from those that are unknown. The objectives included the collection of Pca capensis twigs and extracting them using to Also to fractionate Pca capensis crude extracts, to conduct antiplasmodial and cytotoxicity assays of Pca capensis crude extracts and chromatography fractions, and lastly to conduct HNMR and GCMS metabolomics of Pca capensis chromatography fractions. This was done by collecting the twigs. These twigs were collected from Sekhukhune in the Limpopo province. They were dried at room temperature, ground to fine powder. About 2.5 kilograms of the fine powder was extracted using dichloromethane, which is to 50% methanol in a 1:1 ratio, yielding two fractions or two extracts, polar and non-polar, the dichloromethane one being the non-polar, and the 50% methanol one being the non-polar, or rather being the polar. The non-polar dichloromethane extract was fed liquid extraction using dichloromethane is to methanol in a 1:1 ratio, and yielding two partitions, a dichloromethane partition and a methanol partition. The dichloromethane methanol partition and the 50% methanol extracts were all then subjected to color chromatography using different solvents with increasing polarity. In total, 25 fractions were collected from the fractionation of these three different crude extracts, and all these three different crude extracts, or rather, all these crude extracts and the collected fractions were then subjected to antiplasmodial and cytotoxicity assay. The antiplasmodial cytotoxicity test was conducted using the hypoxanthine incorporation assay, which made use of Plasmodium falciparum in the 54 strain, and the cytotoxicity assay made use of the L6 red skeletal cells. All the 25 collected chromatography fractions and the crude extracts were subjected to HNMR analysis and GCMS analysis. From the crude extracts that were analyzed for antiplasmodial activity, 3 of them displayed the highest antiplasmodial activity. It was the dichloromethane extracts and the methanol extracts. The water extracts did not display the highest antiplasmodial activity. They actually displayed poor activity and a low selectivity index. And to compare these, a decoction was prepared the same way indigenous people would prepare them to compare with the fractions, or rather to compare with the extracts. The decoction was prepared from distilled water, and it also displayed a poor antiplasmodial activity. So it can be concluded that from literature, it has been found that it is only the non-polar extracts in Pca capensis that actually have the highest antiplasmodial activity. But this research has actually displayed the potential of methanol extracts, which also displayed a high antiplasmodial activity as compared to the general notion of non-polar extracts having the highest antiplasmodial activity. And from the fractions, from the chromatography fractions that were analyzed from this study, about 6 of them were actually found to have the highest antiplasmodial activity. In fact, they had the highest antiplasmodial activity even compared to the parent crude extracts from which they were fractionated from. This is an indication that there may be some constituents working in synergy in these different extracts, which can be attributed to the observed antiplasmodial activity of the plant's crude extracts. And also, when GCMS analysis of these fractions were conducted, it was noticed that the bulk of the compounds found in these plants include the aliphatic, about 44%, just sugars as well, some hydrocarbons, and also some acids and alcohols. And it was also noticed that there are some compounds that could not be identified, of which these compounds would be prioritized for dereplication purposes. In terms of dereplication, that is compounds that have not been studied before, and we isolate them and test them for antiplasmodial activity. This is the best way through which we can discover new compounds by focusing on those that have not been studied and leaving out the ones that are already known that have been studied. When the NMR analysis of the crude extracts was conducted, it was found that the 3 extracts which displayed the highest activity showed a similar NMR spectra when they were stacked, and this pattern was tentatively identified to represent the aliphatic class of compounds. But this, of course, was then confirmed when the GCMS spectra of the same crude extracts, which were found to be the most active, were stacked, and the compounds were analyzed, and they were found to represent lupiol and amyrine. Lupiol and amyrine are both triterpenoids, and triterpenoids fall under the class of compounds of the aliphatic, and their presence in these plants, they have also been tested, and literature has been consulted on this. All these compounds, lupiol and amyrine, have actually been discovered to have antiplasmodial activity, and their presence in these crude extracts, which we found to be the most active, can actually explain the observed antiplasmodial activity of the plant. And then when metabolomics was conducted to compare between the NMR and GCMS, metabolomics rather was conducted for both NMR and GCMS data, it was found that it is the NMR model that was able to cluster, rather to separate the different samples according to the observed antiplasmodial activity. The GCMS was unable to separate the data. There were about 2 outliers as well, 2 fractions. And also, when the OPLSDA model was conducted for these 2 NMR and GCMS data, it was found that the model developed from the NMR data was actually the most closest to accuracy in predicting antiplasmodial activity with a p-value of 0.07, while the model developed from the GCMS data ran a very high risk of overfitting with a p-value of 0.74. This leaves us with the conclusion that model that we can most likely rely on for predicting antiplasmodial activity in Pca capensis would be the NMR and not the GCMS. However, GCMS is also an essential metabolomics tool, especially when we are identifying compounds. Following this, we then did a concentration plot to test and see which classes of compounds actually contributes the most in the separation of the observed data, and we discovered that it's the aliphatic class of compounds which have been the ones that contributed the most in the observed separation of this data. The same thing was done with the GCMS data to test which compounds contributed the most in the separation of the data. It was found that beta-amyrine, which is one of the triterpenoids which we discovered to have antiplasmodial activity, was one of the phytoconstituents that contributed to separation. In conclusion, the study has validated the ethnomedicinal use of Pca capensis in treating malaria. The findings of the study highlight the pharmacological potential of Pca capensis as a botanical entity worth of pursuit for new drug development. For future prospects of this research, they would include targeting the most active to further isolate compounds, and this would be done to focus on those classes of compounds that are unknown and applying a dereplicative approach to compound isolation. This will give us the opportunity to discover different compounds that have not been studied before, which can have the potential of having antiplasmodial activity and contributing to the malaria drug discovery. I would like to acknowledge the Victoria Department of Plant Sciences, Department of Plant and Soil Sciences, the Swiss Tropical Public Health Institute, and the Malaria Institute. Thank you.
Thank you so much, Mcebisi. Thank you for your interesting presentation. I don't think we have time for questions, but I would really like to acknowledge your presentation without a visual. It must be very difficult, but well done. Thank you for showing up and doing it anyway. Thank you so much, colleagues, for attending our session and for the interesting questions and comments that you have made throughout the presentations. You've been a wonderful audience. We will now break for lunch. Thank you so much.